Okay, hello everyone. Welcome to the last talk of Snoofer 2022. And of course, we've saved the best till last. So here we're going to have Christopher Currin, who has got quite an intriguing reversal of the normal order of things in this talk, I think. So yeah, do you want to tell us about the strange different way of approaching artificial models and cortical circuits? Please take it away.
Yeah, hey everyone, it's really great to be here. And what I'll be presenting is bridging the gap between artificial models and cortical circuits. There'll be two main sections, and I'm going to focus on number one, is we have a promising personalized medicine approach for understanding and treating individuals with epilepsy. And the second part, I will be talking about our nascent approach for linking spiking neural networks with human brains for the further understanding of both. So it's been a wonderful 2 days of conferencing at Snoofer with people from all sorts of backgrounds and such interesting projects. So it's only right for me to start by saying thank you. It's a great opportunity to be here to present my work. I'm very excited to share what I've done. And a thank you to my collaborators who make me a better scientist, as well as my friends and colleagues, the Vogels group, who make me a better person. And having seen so many spiking neural networks over the past 2 days, I'm going to start with an important reflective question that I think we should ask ourselves, and that is, how close are our models to the brain, and how can we reconcile differences? For example, in spiking neural networks, we principally have two neuron types, inhibitory and excitatory. In biological cortical circuits, we often have many, many more. In terms of simplicity, some SNNs are termed too simple, and biology is often too complex. In terms of tools, we've spent decades building technical, impressive tools to understand both of these networks, but how compatible are they with each other? It feels like sometimes they're talking within that field. Next, in terms of dynamics, spiking neural networks are certainly complex, even if they are simple, and the brain is certainly very complex. But again, how do we relate these complexities and these dynamics that come out of it? It's tough to think. But with all these differences, many current approaches aim to make more complex models, which I think is absolutely great. And we often add extra biologically plausible mechanisms to achieve that. But there's another way too. What if I told you you could have more simple biology? Okay. In this project, I will be demonstrating what we have done making and studying simple biological cortical networks. And I will also touch briefly on the complementary component of building data-driven models. So one of the very special things about this project is that we get to study human cortex. Now, a very simple human cortex, which we view as a benefit because a lot of the really beautiful but burdensome aspects of the human brain's complexity is distilled down. Okay, so what's the recipe for building our very simple human cortex? So we can start with a person, any person, and non-invasively get some skin cells. Okay, these skin cells can be reprogrammed into induced pluripotent stem cells, which means that they have the potential to become almost any other type of cell with the right ingredients. So as neuroscientists, we care about the brain, but we want to make biology that closely mimics our established theoretical models. So we make just two types of neurons. I'm just going to call them excitatory and inhibitory. You can choose the genetic markers. These ones are more like cortical neurons. And yes, these are real human neurons from wild type. I say wild type, it's somewhat mean, but technically accurate for humans without an overt disease condition. But seriously, try getting human neurons from a person without surgery by asking nicely and see how that goes. So here we have an opportunity to grow human neurons without any surgical interventions. Okay. So these differentiated neurons are seeded or grown on high-density multi-electrode arrays. Now, every well has about 26,000 electrodes, of which 1,000 can be recorded from simultaneously at any time. In terms of neuron numbers, this can vary, but we would choose about 200, 220,000 neurons in total, of which between 15 and 25% are inhibitory. And because the MEA, the high-density multi-electrode array, uses extracellular potentials but is high density, we can use a technique called spike sorting in order to extract which neuron spiked when, based on each neuron's unique spatiotemporal pattern picked up by the MEA. So it outputs some spike times for individual neurons. So after spike sorting, this results in us having raster plots, or spike times, for these human neurons. And this is the results I'll be showing in this presentation, where I will have just 2 time points for the purpose here, constrain it a bit. And also each person had their skin cells reprogrammed and differentiated twice. So 2 clones, that's where the term clone comes from in this case. And this in itself is a very technically complex process, and many aspects of this are fields within themselves. And what's making this possible now is that there are new advancements in human-induced pluripotent stem cells in order to grow your neurons feasibly in a dish. And once they're grown in a dish, we can record from thousands of them simultaneously and for many hours at a time. Okay. Now, this overall is very complex and technical, and this process has a very high throughput. So it's hugely data intensive. And when I say it's data intensive, you might be like, well, how big? How much? Come on. So in the data set that I have so far, it's about 986 billion spikes in our data set. But we're still adding more. And these, in our data set, we have human neurons from both epilepsy patients and from wild type. So we really are looking at people with epilepsy. And in fact, for the purpose of this talk, I'm only going to be looking at a small subset of people where they have a gene alteration that has already been linked to epilepsy, and we can start to understand this a bit more. So we have 3 families that we look at, and within each family, there will be a person with epilepsy and a person without, because it's non-invasive. And we have 7 subjects, 78 recordings, 390 minutes of time recorded, 49,000 neurons, about 44 million spikes detected in this data set, and about 400,000 bursts analyzed. So I've told you the stats, but what does it actually look like? Okay, one more pause and then I'll show you. Just to elaborate on X is the time in seconds, Y is neurons, every black dot is a spike, and at the top in blue will be the population firing rate. So here's the activity over 5 minutes, and the cultured neurons tend to have asynchronous irregular firing activity with oscillatory or bursting behavior. And in this case, there are actually quite a few bursts. Now it's informative to compare this raster plot versus another one and say that there are clear differences between people with and without epilepsy. These are from different people, same family, different people. And yeah, you can see clear differences. The one on top clearly has a generally higher activity and more oscillations. But okay, I actually haven't told you which patient had epilepsy and which didn't. And weirdly, the one on top is from wild type, who's a close family member of the one below. And the genetic the gene that's altered in these subjects is SLC13A5. We're just going to call it SLC for the rest of the talk. But this really demonstrates that, well, maybe a 5-minute raster plot isn't the way to classify a difference between people with and without epilepsy. Okay, so 5-minute raster plots aren't the way. Perhaps looking at the dynamics of the network will be more insightful, and particularly the bursting behavior, because a lot of theory says that people with epilepsy tend to have seizures. Seizures are large bursts of activity over time. So that's also could be an important component. So let's look at some measures and metrics of this activity. And the one we're going to start with, of course, is the interspike interval, timing between 2 spikes. It's going to form a foundation for a lot of the future work. And from this, you can look over time per neuron for neuronal activity, such as the average firing rate, the skew, or the coefficient of variation. You could also look as a slice vertically and look at the population. And this could give you the peak rate of activity at the moment in time. Or if you window it a bit more, you can do the cumulative moving average. And this cumulative moving average is a key determiner in our algorithm for detecting bursts, which is also referenced down below. And so some simple measures of bursts include the duration, the number of neurons involved, the amplitude, and then some simple measures, and then some simple measures as well can also be between the bursts as well, like the interval or how often it bursts, aspects like that. Now, part of our burst detection algorithm is also assigning burst properties per neuron in the burst. So we can get a sense of the interspike interval in a burst, how often that neuron contributed to the population activity in total, and then burst spikes as a proportion of the total spikes. Okay. Which I will come back to that point at least a bit later. So the point of all of this measure aspect is that we can extract these metrics of the network's dynamics and pull, summarize, and compare better than just the raw raster plots that are shoved on the screen. Okay, so, and indeed, per person, we have multiple recordings and the multiple clones or differentiated neurons that we can pull from. And here are some examples, multiple raster plots from wild type on the left and SLC on the right, and from the same people, but different clones, different recordings. So a lot of duplicates, yeah. And if we look at the interspike interval, the most humble of the measures, we have stacked histograms to compare. Well, maybe not a clear and obvious difference between them. Maybe the wild type has slightly more activity overall. Okay, but these recordings are from 3 weeks, and previous work has shown that it takes longer than that for these cultures to mature. So let's look at 6 weeks. And okay, it becomes quite striking. You can see a difference. The SLC activity, the interspike interval, overall is just greatly increased. Okay, I'm just going to overlap them just to highlight the difference between the two. Everything's all scaled appropriately. And yeah, there's clearly a difference in the ISIs after 6 weeks. So this is great. Clear difference after 6 weeks. So these subjects, 11 and 12, amazing code names, are from a single family. And we can imagine that differences also occur for other families, right? So let's look at the family, another family over here. And we can see somewhat differences, but maybe not in the direction that we wanted, right? Family 3 wild type seems to have greater activity overall. And so for ISI, there doesn't seem to be a super clear pattern of like, this is the determining factor that can differentiate. Okay, so maybe ISI is not the measure to base our comparisons on. So we just have to find the right metric, right? So here, for example, in each of these graphs is the proportion of burst spikes or number of spikes in the burst out of all possible spikes, sorry, not all possible spikes for that neuron in the recording. So yeah. And these are distributions because it's on a pure per neuron level. So if we focus on the 6 weeks, which is where they are more mature, we can see that the SLC has a large proportion of burst spikes for family 2, but wild type has it for family 3. And just as an intuition for what burst spikes look like, here are 2 examples. This is the first one. It says, well, there's not much activity overall, but when there is a burst, a lot of neurons are involved. So there's going to be a high proportion of burst spikes out of total spikes. But and then on the other side, a very low proportion would be, well, just not much bursting activity at all. So out of the 30 or so metrics that we have, none seem obviously like a key indicator of whether a culture is from a person with epilepsy or from wild type. So what is the right metric to choose? And honestly, right now, we don't know. We're actively working on it. And in fact, one of the more promising avenues is building personalized baseball cards for each person that represents a snapshot of their unique network dynamics. This combines 30+ measures over time that we aim to understand an individual better and motivate for improved treatment options before the usual trial and error approach of current pharmaceuticals. For example, we can look at the interspike interval, which we've seen before, the ISI coefficient of variation, average firing rate, skew, and then also some burst properties like spikes per burst and burst duration and interburst interval, and many more. And we can do this for every person in our data set. So that's just a snapshot. We can do this for every person in our data set. Here are just 3, just as an example. But this is certainly not to say that we're done. The experimental design is certainly an iterative process. So we still have a wish list of things that we are currently doing. And that is, we're going to add more time points. In the fuller data set, we do have more time points. But for the current one that I'm speaking about now, we just chose 2 for demonstration. But we do want also more fine-grained time points, maybe daily instead of weekly. We want longer recordings. We want smarter electrode choice of those 1,000 electrodes you can choose from. Sorry, 26,000 you can choose from. Let's choose 1,000 a bit more cleverly. Let's do some dimensionality reduction on the metrics for some clustering and see which ones are the most important. And then there's some algorithms, class of algorithms called locality sensitive hashing. And this can potentially be used for classification of whether a person is epileptic or not. And then the final one that I'm going to consider more in this talk is a personalized model of dynamics with the goal of understanding the data more, as well as probing our general model assumptions further. Okay, so let's talk about what a personalized model means. And here we call it affectionately a digital twin. The premise is that it's a data-driven model that allows rapid and reproducible probing of dynamics. So of course, there are going to be many open questions. Some fundamental ones are, well, what model do we choose? The large scale and yet also personalized fitting of the data is an open question. And then the interpretation of models in order to conduct meaningful experiments that tell us more about the data and the model. Okay, so how do we create a data-driven model? So different models come with different assumptions, and these different assumptions can lead to different conclusions. So we want models that are informative and explain the data across individuals well enough to provide insight into possible mechanisms. Some examples of model assumptions that can also provide mechanistic insight are the relatively recent conductance-based adaptive exponential Leakey integrated fire model, as well as short-term plasticity models, which consider neurotransmitter utilization and depletion. So both of these can display oscillatory or bursting dynamics, as well as provide experimentation in pharmacological interventions, for example, by having GABA or neurotransmitter depletion. Okay, so from the data, we have measures of the activity that we want to fit. So fitting is hard, but the most promising approach right now is something called simulation-based inference. And there's an iterative filtering to models with desired features. And we don't care about exact spike times. We care more about distributions of the activity. It doesn't have to be exactly the same. Certainly our data from time point to time point is not. Okay. From this SBR, simulation-based inference, come a large pool of candidate models. So this, all these candidate models would fit the constraints that we've iteratively processed down. And there could be different clouds that are slightly different to each other, but we don't quite know how different yet. And that leads to the next aspect of, well, the space of possible models creates predictions about how both the data and the model responds to perturbations. These degenerate solutions in these different spheres, different aspects, could respond differently. And we have the capability in experimental setup in order to apply chemical or electrical stimulations that we can perform to narrow down this model space. And that brings us back to the data and telling us more about how biological human neurons work, as well as the type of neurons that best match the biology themselves. And that brings me to the end. So here's a summary, but please forgive me if some figures were a bit rough, as the last minute habits of generating refined figures a few days before was halted because of a ransomware attack, which has now been rectified. Unfortunately, our work is intact. So we really do look forward to the promising and exciting future directions of this project. So thanks for following me along for the ride. I really appreciate you, your time, and your energy.
Thank you very much, Christopher. We ran over a little bit, so I'll keep the questions relatively short. And I have to apologize that I rather snarkily wrote a question that everyone upvoted to the top, which is, are these networks sentient?
Maybe based on the definition of dish brain, which is not the definition that I would tend to default to. So I would not consider these as sentient.
Yeah, I think I'll agree with that. Michele Nadin asks, is the proportion of inhibitory cells higher than in the real cortex? And also, is the genetic alteration expressed in all cell types or only especially in the two you grow?
Right. So in the first aspect, it's a similar proportion. They are obviously, the brain is highly variable. And even when you do some seeding, the amounts of inhibitory neurons that survive tend to be less than the excitatory ones. So you do get like a drop off a little bit between what the initial proportion you place it is on. But it does, you can do some checks after a few weeks or months, but the check is kind of like you're done with the culture at that point. So yeah, it's a similar proportion. There have also been some really cool recent results with a similar setup where they've changed this proportion to see how the network would respond. And in fact, they look pretty normal with like 80% inhibitory neurons. That's not to say that's what the cortex is, but it's still interesting to look at.
Somewhat relatedly from Bill Podlaski, I'm not sure if I missed it, but what makes these cultures cortical? Is it the neuronal differentiation controlled to get particular neuron types? Or?
Yeah, that's right. The ingredients we sprinkle to differentiate the hiPSCs with the right molecular markers at the end that we check afterwards. Yeah, or what we want and then double check.
Cool. All right. Well, I think I probably better end it there because we're a few minutes over already. And I don't know if Friedemann wants to come on to close out the sniffer, because that was the end of the last talk. So from my point, thank you so much everyone for coming and for making this such an amazing event. Friedemann, do you want to add anything?
Not really, no. Also, thanks from my side. This is it, and I would say this was a lot of fun. Let's do it again next year.
Yeah, I think we'll definitely do it again next year, and let us know if there's any particular ideas about how we should do it again next year.
Exactly.
Yeah.
So thanks, Chris, for this nice talk, and thanks to all the speakers. And one big thanks again to Tim and Katie, again, for helping us with the program selection and all of the moderation today. And we'll be in touch for starting the same thing the next year in a similar format.
Brilliant. Thanks so much. Bye.
Bye-bye.